and welcome to the latest Science Insights from New Scientist. I'm Penny Sarche, joined by my co-host, Rowan Hooper. Hello.
Hello.
On the show this week, we're investigating the latest on the H5N1 bird flu and the prospects of it becoming a human pandemic or not. And we're also hearing some really important news from Italian physicists on the most scientific way to make a classic pasta dish.
That's a brilliant story.
Yeah, I'm really looking forward to this one, it has to be said.
But we're going to start with the latest on gene editing in humans. And what we mean by that is the permanent changing of an embryo's genome such that the genetic changes would get passed on. And it's illegal in most or in many parts of the world. But famously in 2018, in China, there was a geneticist called He Jiankui who used CRISPR gene editing to try to change the genes in human embryos. And they were implanted into women and brought to term, and 3 little girls were born. That, as far as we know, though, the editing didn't quite work as planned.
Yeah, that was a huge shock, and he did go to prison for it.
He did. He had got 3 years. So the reason this is back in the news again, though, is there's a paper in Nature this week looking at this field, because although Hay only edited 1 gene in the embryo, it's now becoming possible, or it will be soon, to edit many, or maybe even dozens or more, to make many edits to genes at once. And that really raises the question of the prospect of doing some really interesting different kinds of monkeying around with the genome. Michael LePage is here to talk about this. Michael, maybe let's start with why you'd want to make dozens of changes in a genome.
Yeah, so there are lots of variations in the human genome, and some of these variants appear to protect against various common diseases like Alzheimer's and diabetes. So in theory, if you edited an embryo so it's got one of these variants, it would confer that protection you see. But the thing is, most of these variants, they only confer a little protection. So it wouldn't be worth doing gene editing just for the protection you get for one. Say it's like a 5% reduction in heart disease or something. But what this paper is claiming is that if you made loads of these edits, so you introduced like dozens or hundreds of these variants, is that this would add up to a really dramatic effect and would dramatically change your risk of getting these common diseases.
So this is what they call polygenic editing, precision editing of the, as you say, dozens or even hundreds of different genes. But technically, how close are we to be able to do that?
Well, according to this paper, we're about 3 decades away from doing what they think would be necessary.
I mean, isn't that a long time, given how we've come so far in 3 decades?
We have, and we can already make edits to several cells at once, but it doesn't work perfectly every time. So that's okay if you're trying to cure some disease in an adult. Say you're editing liver cells. For most diseases, you don't need to edit every cell perfectly. It's okay even if you just do half or even just a third. That's all you need. But if we're talking about editing embryos, and those changes you make, they're going to end up in every cell in that person's body, and then they could be passed down for several generations. So if you're talking about that kind of editing, you need to do it absolutely perfectly, and that's really difficult to do.
Right.
And this is one of the reasons this paper has come under fire from other biologists. They say it glosses over these safety issues and how hard it is to do these things. But to be fair to the authors of this paper, what they are saying is, look, we might have the technology to do this in a few decades' time, and we need to start thinking about whether and how we use it if we do get it.
I mean, it's funny that so the people who are saying that, they've published it in Nature, and then the same journal, the same issue, has another comment piece really attacking that quite strongly, and almost hinting that it was irresponsible for them to publish that. It's funny to have it in the same journal.
Yes, I think this is definitely a very divisive paper, and certainly some of the critics of it view it as irresponsible to publish. One of them described it to me as incredibly speculative. And so, of course, I've mentioned this issue of, can we do it safely? But then the other is, can you really achieve these big health benefits that they're claiming in this paper? That's the other big issue here.
Yeah, and just as an aside, I think it's quite interesting that He Jiankui, when he submitted his work on those gene-edited embryos, he submitted it first to Nature, who rejected it. And we don't know whether they then informed the Chinese authorities about his work or not. But there is some ethical murkiness around what happened there. It's just interesting that Nature involved again in this another controversial paper.
I think his case is quite informative, though, isn't it? Because, he clearly sees himself as a pioneer. He as much describes himself as that. But anyone could have tried CRISPR on an embryo. There's a difference between whether it's possible and whether it's actually effective or safe, right? And that's what we're talking about here. It might well be possible to do this in 30 years' time. Will we really know enough about the genome to do it safely is another question, right?
Exactly. Yes, there were thousands of people around the world who could have edited kids' embryos. There are thousands who could do it now. It's not. Doing it isn't a scientific feat. It's just something that shouldn't be done because it's not safe.
All right. So let's say that you can do these multiple edits technically in the future. Surely then you really would start to see some really dramatic health benefits. Or is that part of the claim, is that controversial as well?
Yes. So what the main paper, which is by Peter Fisher at the University of Queensland and colleagues, what they did is they looked at these big association studies where people they trawl through medical data and they compare it to the gene variants that people have, and they go, oh, well, we found that people with variant X, say, have got a 5% less chance of getting heart disease, and variant Y reduces the sort of chance of getting heart disease by 10%. And so what they've done in the study is they've added up the protection from all these variants, and they said, okay, suppose we edited people and we added all these different variants in, what would all that protection add up to? And they've got some really dramatic results. Okay, so for instance, according to their estimates, if you made 10 edits, you could reduce the prevalence of type 2 diabetes by 60-fold. With heart disease, it could be up to 30-fold for 10 edits. For Alzheimer's, it would be nearly 10-fold for 10 edits, and so on. The only sort of disorder where they didn't find much benefit was major depressive disorder, where you'd only get sort of a halving of the risk with 10 edits. And then so they put all that together and they say, well, if you do sort of 40 edits all at once, an individual's lifetime risk of getting Alzheimer's, schizophrenia, type 2 diabetes, those would all be reduced to less than 0.2% for each one of them. Yeah. So those are pretty big numbers. Those are pretty substantial benefits.
But genetics is complicated, right? And often when you're targeting, to put it mildly, right? often when you target a gene that is involved in something, if you dial it up or down in the wrong cell at the wrong time, it could have all kinds of other potentially really bad consequences, right? And we don't necessarily know what those would be.
Yes, exactly. What we do know is that the variants that they're talking about in this study, these are the variants that have the biggest protective effects on these diseases. But what we know is that those variants with big effects tend to be rare. And then the question is, why are they rare? And they're probably rare because they have, as you were just saying, they have other undesirable effects. That's why they haven't been selected for in evolution. And this is just one of a whole bunch of problems. For instance, in many cases, we can't even be sure that the variant identified in these association studies is actually the cause of that effect that we see in the association. So it's quite complicated, but you can envisage it as imagining that there's a gang that's been shown to be present at a whole bunch of murders. So we know that gang's involved, but who was the actual gang member who pulled the trigger? And the association studies don't tell you that.
Did you come up with this gang member metaphor? I like that.
I think, did Dawkins talked about boats, right? You know that a boat's always winning, but you don't know who's just there along for the ride and who's really doing most of the rowing. Yeah. And when something's really close to other things on a chromosome, you don't know, do you?
Exactly.
Yeah.
And so for gene editing, obviously that really matters. You put in the wrong variant, you get zero effect, or worse still, some undesirable effect. And then, of course, there's also this assumption that you can just add up the numbers. Well, that's not necessarily true. If these things are working in the same way, doubling them up might not double the effect. And there's yet another complication, which is that genes interact with each other. And so making a change in one gene and then you introduce another variant, having the two together, they might interact with each other in a way that cancels out the effect or worse. You're doing this kind of gene editing, you'll be creating combinations that have never been seen in any human before. We don't know what would happen. And then, of course, it also depends on the environment. A variant that is protective in one environment is not necessarily protective in another. So there's this huge number of complications and assumptions that are being made in this study. And the bottom line is, it might be possible to achieve some protection by making multiple changes, but at the moment we just don't know how to do it. We don't know which variants would work and which would be safe and which wouldn't. We just don't have the knowledge to do it.
Talking about how they interact makes me think of an ecosystem and different species. And maybe we need a new field of like ecological genetics to explore all these interactions. The other thing, of course, we haven't mentioned though is eugenics. And they're talking about getting rid of diseases here, but people are immediately going to start thinking about making edits that purportedly could improve someone's intelligence or height or all of these other things. This is really why it's such an inflammatory area, isn't it, of research?
Yes, obviously there are all kinds of ethical issues involved with editing the genes of our children. And to be fair, this paper does go into that. A lot of the paper is a discussion of the ethical issues involved in this kind of approach. And of course, one of the big ones, for instance, is if we're going to do this kind of editing, who gets to decide whether it's done? Is it going to be government saying you've got to edit the genomes of your kids, or is it just going to be left to parents? And obviously that makes a huge difference in the outcome. I mean, imagine Russia and North Korea. What would they kind of impose if they were decided to get keen about this kind of gene editing? And even if you say, okay, maybe this is possible, maybe we can do this kind of gene editing and it will have big health benefits, there's still sort of all kind of questions of, who gets to have it and what are the results of that. So, for instance, if this is something that only millionaires' kids get, you can see this will increase the existing divisions in society. I mean, on the flip side of that, if suppose, the UK's NHS were to offer this to anyone who wanted it, then I think the outcome would be quite different.
Is there a lesson to be learned from embryonic screening? Because we're getting towards a place now where you can score an embryo and guess if it might have a higher IQ or if it might be taller. And most countries have not allowed that. They might start contemplating it if you know that there's a strong risk of genetic disease. But other than a few sort of clinics in far-flung places.
But you could get it done privately, though.
I don't think you can get it done in the UK. I don't think you can select an embryo to be taller in the UK right now.
No, in the UK and many countries, there's very strict. You can do it for serious diseases, but not for things like IQ or anything like that. In the US, you can do that kind of thing. The thing is, though, what you can do with embryo selection is very limited because you've only got a few embryos to choose from. So it's not a sort of the same huge ethical minefield, I don't think, as gene editing, where if you're talking about making dozens of changes, then you're talking potentially about more dramatic changes in the children, and it becomes a much bigger issue. Yeah,
I mean, I do sort of worry that even if it was this utopian ideal where it was on the NHS and anyone could get it, that there's still going to be people without it, and you're then going to start to get a speciation event between the haves and have-nots, and you wouldn't want to mate with someone who's still got bad genes, right?
Yeah.
So it's fraught with dangers.
And I think just to stand back and look at this in the bigger perspective, when we're talking about the health and the intelligence of children, it's not just about genetics. There's so many other things involved, and we already know of so many ways to improve the health and the intelligence of our children. So, from offering family planning to better care for pregnant women to looking after young children better diets, better schools, making our societies more equal.
Things that tech bros don't care about.
Making sure that there are things like no lead pollution in the environment, which obviously has a huge impact on intelligence. And we could even add climate change, that's going to impact the health of so many people. So there's so many things we can do to improve the health and intelligence of our children right now that are so much easier than trying to sort of muck around making hundreds of changes to our genomes. And if that's what we care about, that's what we should focus on.
Here.
Next up, we're going to talk about bird flu, as this week on the 6th of January, public health officials in the US announced that a person in Louisiana had died from bird flu. That's infection with the H5N1 virus. And this is the first known death related to this virus in the United States. So the news has quite understandably stirred fears again about the potential for H5N1 to spark a pandemic. And so to discuss that with us, we're joined by health reporter Grace Wade. Hi, Grace.
Hi.
So H5N1, it's the virus that we've been worried about for years, decades even. Does this news mean we should be even more worried now?
I mean, of the pathogens that have pandemic potential, H5N1 almost certainly tops the list. So should we be more worried? I think we should be cautiously concerned, right? We know there is a version of H5N1 that has been circulating since 2021 and has rapidly spread to flocks all over the world.
Yeah, I mean, you've probably seen the dead birds all over the place as well.
Not actually on the beaches, but some of the footage you see in breeding colonies have been horrendous.
Well, like gannets. It's awful, yeah.
Right. But the reason that infectious disease experts are particularly worried is that H5N1 is remarkably adaptable. So despite being very much a bird flu virus, it has jumped into dozens of mammal species, included foxes, bears, seals, and more recently, dairy cows. And analysis of the virus that killed the person in Louisiana suggests it has mutations that may give it a better ability to bind to cells in the upper respiratory tract, so, your throat and nose. I spoke with Megan Davis at Johns Hopkins University about this. Here she is.
So any change that could make it more virulent or more capable of adaptation into humans or other mammalian species means that there's a greater probability that the virus could kind of take the next steps down that pathway and become better established in people. So that's a big concern. And we're also concerned about the potential for people to be co-infected with strains already adapted to humans, like seasonal flu, and an H5 strain, because that could allow a little bit of reassortment, what we call kind of virus hanky-panky going on, where they can swap genes, and that allows for a bigger shift much more quickly than what we see is these mutations occur, and then they go into the great experiment of different hosts and, different durations of infection. Our more severe cases, right, are infected for a long period of time potentially, and that can also maybe allow the virus to test out new changes to see if they work better.
I do like virus hanky-panky.
Technical term.
But that's how viruses can evolve so quickly, isn't it, Grace?
Yes, Megan says that the virus's traits have put public health experts on high alert, but the general public doesn't need to be on high alert. We shouldn't worry about the possibility of a pandemic yet.
There's been about 1,000 reported cases of H5N1 in people since 2003, and the mortality rate, if you look across this, has been really high. It's about 50%. But that's often exaggerated, isn't it? Because we're only hearing about sort of the severe infections. Those are the ones that get noticed at this stage. And in the US so far, the mortality rate has been much lower in the cases that we've seen.
Yeah, that's right. So last year, 66 people in the U.S. tested positive for the virus, and only one of them, the person in Louisiana, died from it. And that person also had a high risk of severe illness and death. They were over the age of 65 years old, and they also had underlying health conditions. And about those mutations helping the virus bind to the upper respiratory tract, the U.S. Centers for Disease Control and Prevention believe these mutations developed in the person during the course of their illness. So as long as the virus was a dead end with that person tragically passing, those mutations shouldn't have passed on.
So say this does gather pace and it does become more of a concern. Do we have the vaccines we need? Are there vaccines specifically for H5N1?
Yeah, so there are actually 3 vaccines for H5N1 that the U.S. Food and Drug Administration has approved, but they aren't available to the public. Instead, they're kept in the country's vaccine stockpile in case of a pandemic.
Does that mean then that we're in a much better position than we were with COVID? We don't have to start from scratch. There are vaccines and we could just ramp up production.
Yes, we are in a better position than we were when COVID-19 emerged. I think COVID-19 really caught public health agencies off guard. There are other vaccines in the works, including one from the biotechnology company Moderna. Phase 3 trials of this mRNA vaccine are expected to start this year. And the experts I spoke with say it's clear that we are in a much better position with H5N1 than we were when COVID-19 emerged. Here's Megan Davis on this again.
Preparedness comes in many different forms. When we think about preparedness from do we have the countermeasures? Are we on track with development of vaccines? The answer is we do have countermeasures. Right now, they're working pretty well. I think that we have some barriers, though, because of vaccine hesitancy, not just among, dairy and poultry workers, but kind of writ large across the country.
She mentioned vaccine hesitancy there, and obviously that's something we're really worried about, and that could be even more of a problem if we have someone like RFK Jr. as incoming health secretary in the US. And I asked someone else at Johns Hopkins about this. This is Andy Pekosz, and here's what he had to say about it.
I think a lot of the misinformation really comes into areas that I would call conspiracy theories, areas where people are claiming that there's some sort of larger cabal or some sort of conspiracy, right, going on in scientific circles to get a certain point across. Nothing could be farther than the truth. Scientists are about making decisions based on data. And importantly, they're also about making decisions or changing their decisions when the data indicates so.
I mean, but what we've learned from COVID is we've got to be, and I can say that to us as well as science journalists, better at transmitting this information and communicating risks to people, because that is the worry, isn't it? Vaccine hesitancy.
Yeah, and I was speaking to some experts recently on lessons learned from COVID, and there's something there about being really realistic in communication about what a vaccine is for. It's not going to protect you forever necessarily. It's not going to cut down transmission. You might need a booster. I think people went on a journey with the COVID vaccines where people were a bit confused about what they were for, and the goalposts kept changing, and that doesn't seem to have helped. Michael, you did a big piece for us recently on pandemic preparedness, and there was a real thread in that one of the biggest worries is not surveillance or developing a vaccine, but just making sure that we have that public trust and can counter misinformation.
Yeah, I think it's a real problem, and it seems to be growing, especially on social media and the trends we've seen. People were telling me this is now what's worrying me most. I'm talking here about health experts saying this is now what's worrying us most if there's another pandemic. It's not, do we have a vaccine or are we going to spot it in time? It's, are people going to listen to us when we say you should do X or Y?
Right, okay then, let's talk about the big news, and that's cooking pasta.
Yay.
Our colleague Alex Wilkins this week found an amazing paper on the scientific way to make cacio e pepe. That means cheese and pepper, and it refers to the classic and very simple dish of spaghetti with grated cheese and black pepper.
You say very simple.
That's what I thought.
But so this study has happened because we've got a whole load of Italian physicists working in Germany, missing their food, trying home cooking, and they're just getting frustrated that this simple dish is coming out different every time. And being physicists, they decided to really rigorously test all the different methods for making it.
I love it, but for such a simple dish, how can there be that many different variables?
So there's spaghetti, black pepper, pecorino, and the starchy pasta water, and it's about combining those. And it's all about how you combine them to get a different consistency of sauce. And these physicists are phase matter physicists, so they're talking about the phase transition of the cheese and the water mixture. If you get it too hot, or it has the wrong balance of cheese and starch, then you get these lumps in it. They're very upset if they get lumps in the sauce. And Alex called up the physicist, and I was sat next to him when he was on the call. It lasted almost an hour. Wow. It was the most amazing thing. It was the most Italian thing ever, really. The title of the paper is Phase Behavior of Cacio e Pepe and Cacio and Pepper Sauce. And here's a clip of physicist Ivan de Terlizzi talking about the phase behavior.
Most of the times, the starch in the water is not enough to avoid this mozzarella phase. And there is a critical threshold, which in a certain sense implies a sharp transition between two phases. A phase where you have this mozzarella-like clump, which is kind of solid. In some models, you find something like that, and it's called a gelation transition. And the implication in that is that you want to avoid this region. So you have to go above 1% of starch to get a phase, a smooth phase. And then we also add that if you go above 4%, then the sauce is a little bit, it has not a good texture. So just the right amount to stabilize the sauce, but to let a little bit of phenomenology to come out and to be observable. And this is the 1% of starch.
I think by phenomenology, he means just a little bit of cooking magic. He allows that. Honestly, this call, 50 minutes. At the end of it, Alex said, very politely, thank you. I've learned more than I ever wanted to learn about pasta, ever thought possible.
What is the news you can use? Is there a take-home message?
Well, so it's very controversial.
Yeah.
Is that they would add cornstarch to the sauce and thicken it slightly. And that gets the correct consistency, but it has caused, I'm told, it's caused outrage amongst the Italian community there.
This reminds me of our in-house gourmet new scientist, Sam Wong. He once wrote for us about the merits of making mac and cheese by cooking the macaroni in milk so that you didn't have to bother with making a roux. Yeah, absolutely shocking.
It's just like putting pineapple on...
no, don't start. We'll put a link to the recipe for this cacio e pepe. But I want to end with another clip from another one of these physicists, because I just love how deep they go into this, how passionate they are about getting this right. And in particular, how they find that the behavior, the phase transition of the pasta sauce has implications in other scientific fields. And here's the lead author, Giacomo Bartolucci.
For sure, I was very, very fascinated, and I saw kind of the pieces of some theories that I've applied in different contexts, like origin of life or like cell biology.
Origin of life and cell biology.
Origins of life.
Yeah, and look, it's the start of January. This is a contender for story of the year already. Right there.
Everyone else go home. We're done. Yeah. That's it for this week. Thanks to all our guests and thanks to you for listening. Do tell your friends about us. Give us a 5-star review on Spotify, Apple Podcasts, wherever you listen.
And we'll be back next week. Bye for now.
Bye.